evening to all the students. So yes, we are starting with genetics, which is very important. If you can take 2025 paper into consideration, okay, half quarter of the paper I can tell you in biology, it is from genetics. The questions are asked from genetics, combined principles of inheritance and variation and molecular basis of inheritance. You will get 25% in your exam, okay, if we analyze 2025 paper. As such, how you have to go through this chapter, I will tell you. First is principles of inheritance and variation. Very first thing is, okay, monohybrid and dihybrid cross, that is given by Mendel. Okay, take down monohybrid and dihybrid cross. Fine. This monohybrid and dihybrid cross by Mendel, through what model organism he has proposed it. Okay, model organism is P or Pisum sativum. Why Pisum sativum is chosen as model organism? For how many years he has worked on that and all, statement based or assertion reason you will get. Go through proper NCERT liners, and then from monohybrid cross he has deduced first, that is law of dominance, and second is law of segregation. Fine, right? Next, if you see dihybrid cross, the law that is proposed is law of independent assortment. Are these three laws universal? No. Law of segregation is universal, but law of dominance is not universal. There are exceptions of law of dominance, and then comes the concept of exceptions. Okay, just a second. Exceptions. Exceptions of law of dominance include incomplete dominance, codominance, pleiotropy, and multiple allelism. Fine, right? From these exceptions, every alternate year if you see, there will be one question in NEET. Okay, that important it is. All the exceptions, their definitions, genotypic, phenotypic ratios, and the examples under each exception. Okay, and then comes the dihybrid cross, in which law of independent assortment. Here we can expect numerical based questions and application based questions from dihybrid cross. Okay, and then here we see, next is the concept of mutation. Mutation. Yeah, mutations. We have two types of mutations: chromosomal mutations and gene mutations. Okay, followed by this you have Mendelian disorders, and chromosomal disorders. Last but not the least, you have the concept of sex determination. We have three types of sex determination. First type is male heterogamety, female heterogamety, and then we have haplodiploid type of sex determination. Fine. Have you analyzed the complete picture of this? All the segments are important, ma. Everything is important here in this. There is nothing to leave. Mall bio. Done? Got with the framework? First is monohybrid, dihybrid, then laws which are deduced from monohybrid cross, laws deduced from dihybrid cross, exceptions of monohybrid cross, then comes the concept of mutation, then disorders, pedigree analysis, sex determination. This is the complete picture of principles of inheritance and variation. Okay. Now we will get started with this. First thing, before getting into the concept of Gregor John Mendel's experiments, early agriculturists, okay, who are present in around 8000 to 10,000 BC, they have already understood the concept of variation in detail. Okay, they have already understood the concept of variation, like they have said it clearly that variation is because of sexual reproduction. As such, they have produced the dominant breeds like Sahiwal cow of Punjab. One such example is Sahiwal cow of Punjab. Okay. So, after early agriculturists who have deduced that variation is because of sexual reproduction, it is Knitt and Gauss who has worked on artificial hybridization. Who are those scientists, beta? Knitt and Gauss. After them, our Gregor John Mendel has come into picture. Okay. He has worked on the pea plant for 7 years. He has worked on pea plant for 7 years. From 1856 to 1863, and the model organism he has taken is, yeah, model organism is Pisum sativum. Fine, right? Why he has chosen Pisum sativum? Why garden pea? Why Pisum sativum? Why garden pea? If you look into the reasons why garden pea, because it is an annual plant where it completes its life cycle in 1 year, one single year. That is one major reason. Second is it shows clear contrasting characters. You take any character in the pea plant, there are so many characters, but Mendel has chosen 7 contrasting traits. Trait or character is one and the same. For example, you have black hair. Okay, hair texture is one character. For example, I am telling you, your hair texture is a character. Okay. Now, that character can have 2 variations. It can be 2 or more. Okay. Let us suppose black and brown. Okay. Those are variations. If that character is transferred to the next generation, then that is addressed as trait. Okay. Mostly character and trait are used as synonyms, but the one which is transferred to the next generation is addressed as what? Trait. Here you see it has what? Clear contrasting characters, and it shows self-pollination with true breeding. Artificial cross-pollination is seen, and this shows stable trait of inheritance. These are 5 reasons why Mendel has chosen pea plant. All the reasons are important, I am telling you. We may expect a statement based question. All the reasons are important. Once it is done, let me know. If you are not getting anywhere, you can ask me. Done? Here we go with next. Now, what are those 7 contrasting traits that he has taken? Remember, Mendel has taken how many characters? 7 characters or traits. How many variants or variations? 2 variations each, so on total variations are 14. First is, let us suppose seed shape. Okay, seed shape. Seed shape, round is dominant and wrinkled is recessive. Fine, right? This is seen on what chromosome? 7th chromosome. Okay, and next if you see seed color, okay, seed color and flower color is present on the chromosome 4. Seed color and flower color. Flower color dominant is purple, recessive is white. Seed color dominant is yellow, recessive is green. Pod color is seen in 1st chromosome whose dominant trait is green, recessive is yellow. Pod shape, plant height, flower position is on 5th chromosome. Okay. Dominant variant of pod shape is inflatted, recessive is constricted. Dominant for plant height is tall, recessive is short. Dominant for flower position is axile, recessive is terminal. Done? Remember ma, of course in your NCERT might be or your exams as well, if you want to see in your IP also, most of the times what is asked, the example most of the times it is given is tall and dwarf, yes or no? But then for your exam any type of variant can be asked. You have to remember all the dominant and recessive variants for the 7 characters for sure. Okay. Of course, chromosomal position I have given you, that is exceptional. Okay, it is up to you, but then it is your responsibility to remember all the dominant and recessive traits. Clear till here? Yeah. Next, if we see Mendel's monohybrid cross. Okay, any cross, even in your IP exam when you are presenting, it should start from parental generation now. Okay, for here you see monohybrid cross. First, to start the experiment, you should know about artificial hybridization, where we have already discussed in sexual reproduction of flowering plants. What happens is 2 plants are taken, okay, they should be pure line dominant and pure line dwarf, tall and dwarf. But we have learned that pea plant is what? Bisexual plant. So, of course, anthers should be emasculated. So, first in that bisexual flowers, anthers will be emasculated, it is bagged, and then respective pollen grains will be dusted on it, and then it is rebagged. That is how we are conducting artificial hybridization. And then gametes, only one type of gamete here you will get, that is capital T, here another type of gamete, that is small t. In F1 generation, if you consider first filial generation, we will call her, you will see only dominant phenotype. Dominant phenotype and recessive and heterozygous genotype. Genotype is heterozygous. Okay, genotype is heterozygous. Now, selfing of F1 generation. Okay, selfing of F1 generation will give you second filial generation, where phenotypic ratio is 3 will be dominant, 1 is recessive. Okay. Genotypic ratio 1:2:1. 1 is homozygous dominant, 2 are heterozygous dominant, 1 is homozygous recessive. Till here it is clear, right? How you got to know the genotype of the tall phenotypes?
Test cross.
Very good. Test cross comes into picture. Okay. What is test cross? That fusion between the F1 generation and recessive parent is addressed as what? Test cross. The cross of F1 generation with recessive parent is test cross. Fine, right? So, if at all in test cross the genotype is dominant, capital T, it is there once. If the genotype is dominant, which is homozygous, when it is crossed with recessive parent, all will be heterozygous dominant, no? So, phenotype will be all dominant. If it is heterozygous dominant, you will get 1:1 ratio. That is how you will differentiate between the genotype. That is how you will get to know about the genotype of the offspring. Okay. So, this is test cross. What is back cross?
Any parent.
Yeah, F1 generation, if it is crossed with any of the parent, that is addressed as back cross. Both test cross and back cross will give you 1:1 ratio only. Okay. Now, so test cross and back cross is clear, right? You will get application based questions with test cross and back cross. Remember that point, test cross is helpful for what? Identifying the genotype of which generation? F1 generation. Monohybrid cross is clear to you. Now, from monohybrid cross, Mendel has proposed two laws. One is law of dominance, second one is law of segregation. What are the definite points of law of dominance? He said that every character or trait will have discrete units called as factors. Write down, every character has discrete unit of factors. And second point, factors occurs in pairs. Factors occur in pairs. Fine. In this pairs, one allele is dominant. This masks the effect of recessive allele. These are the points which are given out from law of dominance. Quite a times, if you see in your NEET previous year questions, multi-statement based questions have asked only on the law of dominance. And law of dominance is not universal. This is not universal. Okay. Here comes law of segregation. Law of segregation comes into picture at the time of gamete formation. Or else you can directly take out during gamete formation. This is also known as law of purity of gametes. This law is also known as law of purity of gametes, because when two alleles occur in pairs, there will not be any mix or blend of the character. Okay, there is no mix or blend of alleles. Fine, right? There is no mix or blend of alleles. And there is no mix or blend of alleles, and this law is universal. We have addressed that law of dominance is not universal. Then we have to discuss what are the exceptions also, no? exceptions of law of dominance. Okay, listen here. First is incomplete dominance. Incomplete dominance is a phenomena. Okay, this is a phenomena which is observed in snapdragon or antirrhinum. Okay, wherein the dominant flower color is the character beta. Here dominant allele will not completely mask the effect of the recessive allele. Okay, but according to law of dominance, what is he saying? One allele will completely mask the effect of recessive allele, but that is not true with the case of incomplete dominance. Second is codominance, where two alleles will show equally dominant features. For example, if you see in blood grouping, the combination of alleles of IAIO and IBIO, both alleles will show the effect, which is codominance. Okay, and you can take the example of starch synthesis also, which is capital B small b, heterozygous condition. Both the alleles will have the effect, which is codominance. Sorry, starch synthesis you can consider it for incomplete dominance, not codominance. For codominance, only example you can take is blood grouping.
Color of cow, black patches.
That also you can take, color of cow, and you can take the example of grains with spotted and dotted grains. Okay. Next is pleiotropy. According to Mendel, one gene has two alleles, right? Okay, one gene will code for one character. Okay, here according in his terms it is factor, now we are calling it as genes, that is only the difference. According to pleiotropy, one gene will show multiple characters or multiple effects. One gene is showing multiple characters or multiple effects, that is pleiotropy. So this is one more exception. Last exception for Mendelian law of dominance is multiple allelism. Yes, multiple allelism, where one character according to Mendel is controlled by two alleles, but here one character or trait is or one gene is having many alleles. That is one example of what? Multiple allelism. ABO blood grouping is again the example for multiple allelism. Okay, clear with the exceptions and laws? Test cross is clear to you. Now, incomplete dominance. Incomplete dominance is first given out by Carl Correns. Carl Correns. He has worked in the on the plant snapdragon, antirrhinum, or Mirabilis jalepa 4 o'clock plant you can take. Here flower color is the character that is considered beta. So the dominant flower color is red, recessive is white. Okay, and capital R, small r are the gametes that are considered. In the F1 generation, when capital R, small r is there, okay, in the heterozygous condition, what should come into picture according to Mendel's first? Red. But here what is there? Pink.
Neither red nor white.
Yes, it is capital R, small r condition. Okay, so in the F2 generation, if you see the genotypic and phenotypic ratio and phenotypic ratio is 1:2:1. Genotypic and phenotypic ratio is 1:2:1. Clear till here? Any doubt anywhere? No, right? Yeah, this is about incomplete dominance beta, where here there is no blend or mix of characters. Dominant allele is not completely masking the effect of recessive allele, because of which you are getting the pink color. Okay. Next, in multiple allelism, okay, multiple allelism wherein multiple alleles will come into picture. Okay, single gene. If you take the character of ABO blood grouping, you take it has how many genes? One gene?
3.
No, I is the gene. But it has how many alleles? 3 alleles. It is IA, IB, IO. Okay. So as of now, you remember this is the example with one gene having multiple alleles. Tomorrow we will see how many number of phenotypes and genotypes are possible. We know of course 4 phenotypes: A blood group, B blood group, AB blood group, O blood group. Okay, but we have 6 genotypes on total. Tomorrow we will exercise that tabulated form. Clear? So this is the concept of what? Multiple allelism. Now we will look on to the pleiotropy. Pleiotropy is a phenomena wherein one gene will show multiple effects or many characters. Or effects. You see starch synthesis. Starch synthesis can be expressed in 3 forms. One is homozygous dominant, next is heterozygous dominant, and next is homozygous recessive. So homozygous dominant are large starch grains with large starch synthesis, high amount of starch synthesis. Capital B, small b, it is of intermediate starch size and intermediate starch synthesis. Showing incomplete dominance, yes or no? Capital B is not completely dominant over small b. If it is completely dominant, you have to get large starch grain. But here it is of intermediate size and intermediate synthesis. And small b is completely small. Small starch size with small starch synthesis. Clear, right? So this is the example. Intermediate starch grain. So this is the example of what? Incomplete dominance. And next one more example we can take. That example is phenylketonuria. Which is PKU. Phenylketonuria. This is a disease which is occurred because of one aromatic amino acid, that is phenylalanine. This phenylalanine has to get converted to what? Tyrosine. Then it will be showing you proper metabolic activity. Okay. This is converted by the enzyme phenylalanine hydroxylase. What is this enzyme?
Phenylalanine hydroxylase.
Yeah. Phenylalanine hydroxylase. Phenylalanine hydroxylase. Absence of this one enzyme should inhibit this activity, right? It will inhibit this activity where phenylalanine gets stored in the form of phenylpyruvic acid. Yes, in cerebrospinal fluid. This phenylpyruvic acid will give multiple effects like mental retardation.
Albinism.
Yeah.
Albinism is due to absence of tyrosine.
Yes. Mental retardation, albinism, frequent micturition, these are the multiple effects that are shown just because of the absence of single enzyme. Understood? So phenylketonuria is one more example. Incomplete dominant, this starch synthesis is one example of pleiotropy. One more example is depigmentation that you will see in Drosophila, skin depigmentation. Okay, these are the examples of pleiotropy. So what all we have exercised today from the beginning, complete picture of the chapter. Okay, second thing is monohybrid cross. Third is laws that are derived from monohybrid cross, exceptions of law of dominance. Of that only 2 we have discussed in detail. One is incomplete dominance, second codominance, 3 we have discussed, right? And third is second is multiple allelism, fourth is pleiotropy. We are behind what? Codominance is yet to be discussed. Codominance, dihybrid cross, law of independent assortment, polygenic inheritance. In the next class we will see along with numericals. Okay. So that's all for today's session. Thank you. Any other doubt you are having? If you are having, you can ask. Mm.